everyone. Good afternoon and Happy New Year. Thank you for attending our first Core Technology Seminar for 2025, sponsored by the Lurie Cancer Center. Our speaker today is Pamela Focia. Dr. Focia is the Structural Biology Facility Manager and Research Associate Professor in the Department of Molecular Biosciences and the Department of Pharmacology at Feinberg School of Medicine. Dr. Focia will be discussing technologies at the structural biology facility that are being applied to cancer-relevant research. So please join me in welcoming Pamela.
Thank you very much, Sarah. Thank you, everybody. So I've been at Northwestern for 24, going on 25 years now, and in about 2002, the structural biology facility was came to life. And I've been working with Alfonso Mondragon, our structural or biology facility manager, faculty director for all that time. Our mission statement is shown here. I'm just trying to get rid of a little Zoom meeting here. I'm not going to read this to you, but I put in bold a couple of things that I want you to really pay attention to. Oh, thank you. I was doing that so I could see better. Okay, thank you for that, Milo. So I've put in bold a few terms that I want you to pay attention to, because my goal today is to tell you about our facility, but also get you excited about structural biology. So we primarily use the techniques of crystallography and cryo-electron microscopy, and we're trying to discern the structure-function relationship between macromolecules. Our facility also strives to build a community of structural biologists that keep in touch with each other on emerging trends and technology. Okay, so our mission statement, these slides will be available to people if they want them later on. I want to introduce our staff. Alfonso, I mentioned, began the structural biology facility with me in 2002. Our EM managers are Ao Cheng, who deals with the screening microscope mostly for cryo, but we're collecting nice data on that microscope currently. A new fellow will be joining us, James, coming to us from Genelia Farms, who will run our new Krios facility that's going to be up in Evanston. And then Jason, I apologize for this picture, but Jason's a really nice, fun person. Both Al and Jason's photos I took as out of candids from a cancer center photo shoot. I didn't have headshots of them to share. But this is our team. And we'd like to meet you and learn more about what you want to work on. So there are many ways to study your protein or your macromolecule of interest. We always have to purify something. You'll learn about the molecular weight. You can learn about mass versus charge for fragments of your protein using mass spec. You can learn something about the function of the protein by doing kinetic assays, or its thermal properties, its stability under heat by using CD. But all of these techniques, so anyway, the techniques I just discussed about learning about things about your protein, they're all very important technology. They help tell the entire story of your macromolecule. So we want to use all of those techniques, but they're indirect techniques compared to what our techniques in structural biology will bring to you, which is that you will actually essentially get to see your protein macromolecule and from my title, up close and personal. I really hope that this can get fixed. Okay. Why might you want to use our facility? As we all know, structure defines and dictates molecular function. So there are mechanisms of many important biological processes that have been revealed by analyzing structural details of important cancer targets and other drug discovery targets. You might want to do some kind of drug discovery, drug design on the project that you're working on. And getting to see your inhibitor bound to your target protein or macromolecule is very valuable. We really get to know our structures, our proteins, our molecules at the atomic level by studying it by structural biology. Okay, our locations, I think it's going to be okay now. Our locations are shown here In the Terry building, we have a small lab with just a couple of instruments. Most of our instruments are on the Evanston campus, unfortunately. So they're all listed here. We have in-house electron microscopy in Evanston. I understand that a microscope is coming here in the coming future. We also have crystallography sample development at both campuses, but we collect all of our data out at the synchrotron at the advanced photon source at our beamlines. The key services that we provide are shown here. We supply all of this instrumentation that's very expensive for the average lab to afford. So that's part of our main key service is providing this instrumentation, but we also take care of it. We keep it running properly. We keep it upgraded. Our facility managers are all experts in their fields, and we're available for consultation. There's a potential for having one-on-one training, one-on-one meetings rather, to start your project to see if it's feasible. And there are many levels of service available. If you want to be trained to do things yourself, we can do that. If you want some feasibility studies to be done, especially on the microscopes, to see if your protein or your macromolecule is of the right size or monodispersity to be able to do that technique, we can run those samples for you. And then you can decide if you want to get trained further and do the work yourself. And that's available on both of the existing microscopes and will be available on the new Krios that's coming in March. So for crystallography and EM, we can provide one-to-one training if you decide that you want to pursue this technique and put this tool in your toolbox for your own use. Some people just want to know the answer. Some people want to know the process and want to get involved in the experiments. For computational drug design, and discovery, we have all of the software. We have much of the software. There'll be a list of software later. But I also wanted to point out AlphaFold is very in vogue right now. Yes. Oh, did you have a question? No, you're just stretching. Sorry. We actually have an AlphaFold server in our computers. So there are public servers that you can use, but they have time limits. So if you want to do really big projects, we can provide that resource for you. We also have molecular graphics for analysis and presentation available. Here's a list of the microscopes and their cameras that we have, or the EM stuff. There's also a sample prep room. It's the state-of-the-art direct electron detectors for the Glacios 200 kV scope and for the new Krios that's coming in the spring. We recently moved into a new humidity-controlled sample prep room in Silverman Hall. And that's really important for cryo-EM because ice and water contamination is a big problem. So we have things to prepare your grids. We have Vitrobots, which freeze your grids with your proteins, macromolecules on them. And then we have a new sample storage system, transport system coming soon that will help get samples from this campus to the Evanston campus. This is what the new microscopes look like. They kind of look like refrigerators. The problem stopped happening, Sarah. So thank you. So the Glacios is what's been in operation for about a year now, and the Krios will be coming, as I said, in the spring. But behind the curtain, this is what they look like. They look like what electron microscopes used to look like. So they've become more user-friendly. They've taken a lot of the place where mistakes can happen, especially with naive users, out of the equation. So it's much easier to load. Now we can load 12 samples at a time. On our old microscope, you had to load one sample at a time, and then you had to wait an hour for the vacuum to come down before you could proceed with your screening even. So this microscope will load 12 samples. If they stay in there overnight or for several days, they'll all be kept safe. So let's switch to crystallography for a while. I wanted to point out in the background of most of my slides, I have what is referred to as electron density. This is electron density from a crystallography experiment. There'll be other density in the background for EM experiments. And I just thought that would be kind of fun. So for crystallography, we have 2 crystallization robots, one on each campus. The one on the Evanston campus allows you to work on membrane proteins in the lipidic cubic phase and in bicels. We have a series of commercial crystallization screens available if you'd like to try them. You don't have to buy them. You can just use ours and give them a try and see if you get any crystals from that. We have something called a dragonfly that allows you to do optimization screens. If you found a lead crystal in your commercial screen, then you can refine those conditions by creating your own screen that's a little bit more focusing on those lead crystals. Microscopes, so we have these UV vis microscopes, and that's to help you figure out if your crystal is a protein molecule or a salt molecule. So we rely on your protein having some tryptophan, so you'll get a fluorescence signal. We do a side-by-side vis and UV picture of your tray, and you can see if those crystals light up, they're protein. We also coordinate access to our beam lines at the advanced photon source that I'll go into in a second. Right now. Okay, so this is the advanced photon source. It's located about 30 miles south on I-55 from here at Argonne National Laboratory. This is a map of the synchrotrons around the world. There's a few in the United States, a bunch of them in Europe, and then Australia, Brazil, Japan. The advanced photon source underwent a very intense upgrade over the past year. We're still recovering from it there. I didn't mention this before, but I work part-time at the Advanced Photon Source at our beam lines, as well as in the structural biology facility. And while we were in the dark period, as they called it, when we were doing the upgrade, we were sending crystals over to Europe and to New York. We sent crystals to France and to Sweden, to the UK and to New York for that period for all of our users, so that their research wouldn't be interrupted during the upgrade. This is a list of the instruments that we'll be putting online. Some already existed, some are new for our beam lines following the upgrade. So we're going to have a 500 times brighter X-ray beam, and we're using now these what are called continuous readout detectors. They collect data without closing a shutter. They just rotate the crystal and collect data, much like that movie you saw at the very beginning of the talk. Also, Northwestern's a founding member of the Life Sciences Collaborative Access Team, which we call LSCAT, and that's where we go to collect our data. The structural biology facility coordinates the beam time at LSCAT. We do the scheduling, we take care of all the safety forms, all the after run summaries. We have beam time once or twice a week over 9 months of the year. That's because we have 3 months on, 1 month off, 3 months on, 1 month off, all throughout the year, because they have to work on the machine on those months off. You're allowed to go on site for collecting your data if you'd like to have the synchrotron experience. Remote data collection is also available. Sit at home in your pajamas, sipping coffee and collect data. We handle all the shipping of the samples from your lab to LSCAT. And we can also arrange with staff of LSCAT to collect your data for you and process it if you wish. And the beamline scientists are also always available for consultation. So here's some of the equipment inside the hutches. These are referred to as the optics. This is just a schematic view of the layout of our beamlines. We have one beamline going straight through, and then two coming off at angles here to these two hutches. This is referred to as a monochromator. So the synchrotron is a circular device where the electron beam is being bent by magnets. And then coming off at trajectories are the X-ray beams, but they still need to have the beam tweaked a bit and bent and put in the right place for us to be able to collect data. This is some of the equipment inside the hutches. This is a crystal right here on a pin. It's frozen. This is the cryostream. The X-rays come out of here and they go this direction through your crystal to a detector. The detector is shown right here in this image. This blue gadget here is our crystal mounting robot. And it will put the crystal on the goniometer here from the storage positions. If you're collecting data remotely or even on site, this is the interface that you'll see. You'll see your crystal in a nice camera where you can center it by clicking on it. This is where your samples are located inside the pucks inside of a liquid nitrogen storage vat. You'll also be able to see the cameras from remotely. You can just decide which camera you want to look at if something might be going wrong and you wanted to try to understand it. This is again that diffraction image, this experiment rather. So we're rotating the crystal in the beam in order to capture data from every orientation of the crystal. So let's switch now to our computational resources. We basically host and maintain all of the software necessary for the major structural biology structure determination methods, crystallography, NMR, and cryo-EM. There's also software for analysis, predictions, or doing simulations, modeling. I mentioned AlphaFold earlier. We have a graphics room that has some big monitors and very fast computers and stereo glasses. And although you can do a lot of work on your laptop, it's really fun to sit in front of a dual monitor display while you're working on building your model with your stereo glasses on. And I encourage people to do that if they ever want to see it even, or if you're doing some of your work there, it really makes the work fun and faster even than doing it on your laptop. We have a very powerful cluster that you can use to run long experiments for whatever types of studies you're doing. We have a bunch of GPUs, which are important for cryo-EM data processing, because the data are just so immense that you need these extra strong computers, extra fast computers rather, for that process. We have the resources for molecular docking studies. And our facility manager, Jason, he is always available. I know there are other computational facilities available that serve the entire university, but I don't have any experience with them, I'll be honest. But I know that Jason, if something's going wrong, Jason will drop everything and fix it right then and there. And Jason is also available for consultation in your own labs. He tends to travel to both campuses. He works at the downtown campus with the structural center for infectious diseases. And he also goes to labs that just need help, and he just charges for his time. He fixes things in 5 minutes, so it's not a big burden financially. But Jason is very helpful and very knowledgeable. Most of our services, except for the stereo viewing on the big workstations with the big monitors, are available remotely from anywhere you want to be. This is a list of some of the representative programs. Also a summary of the types of computers we have now. We're in the process of trying to get even more disk space. We have very fast connections to the advanced photon source and to the electron microscopes. And this is just representative software. If there's any software that is not something that's very expensive, we'll get it for you. We'll install it. It's just that if we do have to spend money, you have to share it with everybody. But that's all something that can be figured out. So I'm just going to start now comparing crystallography and cryo-EM. There's been a lot of talk that cryo-EM is the end of crystallography. But what's really happening is a lot of people doing crystallography are now doing cryo-EM also. There are a lot of things in common between the two techniques, especially at the stages of model building and the computational stuff and refinement. So they share a lot of things in common. Crystallography has been called cryo-EM's big brother. It had a head start. It's over 100 years old. There was an anniversary a few years ago for the 100th anniversary. Many Nobel Prizes. Cryo-EM is younger, and as their technology improves, the resolution of the experiments is getting better and better. And it's been referred to as the resolution revolution. And that was where people were thinking, well, that's the end of crystallography. Everyone just wants to do cryo-EM now. But there are some cases where you can't do it, and there's some cases where you might not want to do it. But you want to have both of these techniques in your toolbox. And the technology for both techniques is continuing to improve. And that's when things really take off, is when the technology improves. The detectors improve, the computers improve, the sources of the X-rays or the electrons improve. And that's what really is driving the science. Okay, so I mentioned that there are some cases where you are not able to do cryo-EM, and that's based on molecular weight. So this graph shows you that NMR tends to be very successful in very small proteins under 20 kD. Protein crystallography is very successful, but as the molecular weight gets larger and larger, it's not quite as successful. We don't have as many structures, and these are structures that have been deposited in the PDB. I'm sorry, I forgot to mention that. So with EM, there's a size limit also. Your molecule has to be big enough. So right now, the cusp is 100 kilodaltons. If your protein is less than 100 kilodaltons, you may have trouble with EM due to the signal to noise ratio. There's tricks you can play though. But as this little summary says, there are a lot of proteins in the human genome that are 75% that are less than 50 kilodaltons. So those will not be tractable by cryo-EM. They'll still need crystallography and NMR. And then this is, let's see. Okay, so that's by size. So this is structures deposited in the PDB versus time. And as you can see, crystallography had quite an exponential growth, and it seems to be plateauing. NMR in blue also seems to be plateauing, and it's never been quite as high as crystallography. And cryo-EM is really taking off, but you can see that only started in 20 years ago or 10 years ago, where it became exponential, but it's still growing. Another issue with these techniques is molecular weight, of course, as I already mentioned, but conformational flexibility. So for crystallography, conformational flexibility is a problem that it makes crystallization more difficult. And there are parts of proteins that are flexible that we don't see in our electron density maps for crystallography because they're moving. What we see in crystal structures is those things that are very well ordered. NMR is when you get an NMR structure, you actually get an ensemble of structures because they take into account that things are flexible, but that's also limited by size. And cryo-EM, you have a little bit more leniency with flexibility, but also a lot more in molecular weight. Larger and larger complexes are possible with cryo-EM. You can see also that there's an overlap area between the two disciplines. I mentioned that there are some tricks with stabilizing structures. That would be like adding a ligand or an inhibitor that may make a protein stay in one conformation because it's holding on to your ligand, which would help with crystallization. There are tricks also for cryo-EM. If your molecule is not large enough, you might be able to fuse it with another molecule, like maltose binding protein or something like that. And then your particle will be bigger and you can do cryo-EM, and you just don't pay any attention to the maltose binding protein part of your density map. Well, of course you have to submit it to the PDB, but what I mean, for your project, you can concentrate on your own protein. A flow chart here now to compare these two disciplines. The crystallography workflow: you purify your protein and grow a crystal. You put the crystal in the X-ray beam. This is where you might have to go back. You might find that you don't have diffraction. The diffraction is not very high resolution. And then you'll go back and you'll iteratively try to improve your crystals and go back to getting some X-ray data and deciding whether or not you can proceed down the workflow. Once you have your electron density map, and you're going to do then what we call model building and refinement. And that's another place where you may have to circle around. You'll do several iterations of building your model into the density, refining it using computer programs, seeing what the result is, going back and calculating a new electron density map and making a few more changes until you reach convergence. After that, you analyze and you publish. In cryo-EM, we have a pretty similar workflow. You have your sample, and you make these grids. You freeze these grids in liquid ethane, put them on the microscope, and look at your images. You might find that your images look terrible and you need to go back and try different parameters when you're making your grids. So this might be an iterative cycle of trying to get your grids better. You may have to go all the way back to your sample preparation or even your protein preparation in order to improve things so that you can get good electron microscopy data. And then you will make a map, an electron density type of map, model build into it, look at your analysis, analyze your structure, but you'll have to go back a few times to look at your map, make new maps, change your model a bit, see the results, and this will be another cycle until you reach convergence. So the two bottlenecks in both techniques that I mentioned that came out of those workflows are preparation of your molecule, which is the biochemistry and the purification, but also the sample preparation where crystallization is a bottleneck. You can't always get crystals. You may have to change something on your protein to get crystals. You might have to add some detergents or something like that, other cofactors, other conditions. Cryo-EM, the bottleneck is the grid preparation, similar to crystallization. And again, this just leads to you having to go around and around and improve things before you get the sample that you need to get the data that you need to proceed. So I'm going to tell you about a couple of really short stories about a couple of eureka moments I was able to have in my career. That'll be when I was the first person to get to see something for the very first time, something that had been thought could be something that was unexplained, and it was very, very fun. Some things can be expected. Sometimes they're just a complete surprise. In large projects where you've got multi-proteins or a multi-step reaction, you might do multiple structures of the same protein so that you can see the steps along the reaction pathway. And then structure-based drug design also is an iterative process because you work with your chemist comes to you and says, I have this great inhibitor of this enzyme. Can we get a crystal structure of it or an EM structure of it? I want to see where it's making its hydrogen bonds and so on. And then once you see that structure, the chemist will say, oh my gosh, I can add a functional group here and it's going to bind so much better, and so on and so on. And that's really fun for the chemist and the structural biologist, who might be the same person. So in my postdoc, I worked on a purine salvage enzyme from a parasite called Trypanosoma cruzi. It was a hypoxanthine phosphoribosyl transferase. It's a protein that does a reaction with two substrates and has two products. In this family of enzymes, and there were a couple of residues in an area that was termed the long flexible loop because it could never be seen in a crystal structure. So it was known to be flexible. It moved around during crystallization, so it wasn't part of the crystal lattice. And no one ever saw it, and no one ever knew what this serine tyrosine dipeptide that was 100% conserved across all organisms, what was it doing? So we were able to use one of the substrates, and one of the substrates was an analog that didn't have the reactive nitrogen on it. So we were able to trap these two substrate analogs in the active site. And in one of the two molecules in the asymmetric dimer in the crystal structure, the loop was actually closed. And lo and behold, when we saw this structure, here's the substrate analog where the nitrogen is not at the 9 position. That would be attacking the phosphoribosyl pyrophosphate right here. You can see that in kind of grayed out there's that tyrosine. It's blocking exactly where that reaction needs to happen to sequester it from water. And it just made so much sense all of a sudden to see that. So the second project I worked on when I was here was a signal recognition particle proteins. There are two GTPases that are structurally homologous, one in green here and one in blue. You can see them in the other photos. But what they do is one of them attaches to the ribose that has a nascent chain coming out of a protein that's destined to become integrated into the membrane or pass through the membrane. It's called a signal sequence. That blue molecule attaches to the signal sequence and then finds its partner that attaches to the translocon of the membrane. Those two proteins had also been shown to stimulate each other's GTPase activity, but no one knew why. And the structures of each of those two molecules were determined several times, but not bound together. No one could seem to get them to stick together. And again, we used substrate analogs to make that happen. We used a GTP analog called GMP-PMP, or PCP rather, GMP-PCP. Now, this is a movie from a friend lab that we co-published on these structures. But as you can see, they're very symmetric to each other. What might be harder to see when it's moving around is that the two GTP molecules actually come into contact when these two molecules are brought together. And they actually form a hydrogen bond with one another. So I think that they are involved in that mutual stimulation of GTPase hydrolysis, or GTPase activity and GTP hydrolysis. Okay, so now switching to what do we mean when we talk about resolution? So resolution is basically how much detail in which you can see atomic positions. And that gives us confidence when we're building our models to know things are in the right place, basically. So this is just showing a phenylalanine, and you can see that you could tell this is not an alanine by looking at the density. It's something big. But it's not really clear exactly what it is until you get to higher and higher resolution. At 1.2 angstroms resolution, there's actually atomicity in the phenylalanine density, and you can see a hole in the middle of the ring, and that's due to the pi electrons delocalizing around the ring so that the center of the phenylalanine ring is electron deficient, while the ring itself that's where all the electrons are. Here's a proline that was from a structure that was determined at around 1 angstrom resolution. And I wanted to point out again the atomicity. You can see these distinct carbon densities here. This is an oxygen from the carbonyl of the main chain. Oxygen has more electrons than carbon, and as you can see, there's more electron density for the oxygen than there are for the carbons. But also right here, that's the peptide bond, which has a resonance structure. So these electrons are also delocalized across that peptide bond there. And we thought that was kind of cool. At very high resolution, again, you can also model, and this is in crystallography. I'm not sure if this is done in EM yet, but you can model multiple conformations of amino acids in your protein. And I have to say, I heard George Manasov in the audience, and this is his work right here. So crystallography and cryo-EM often go hand in hand. Over the years, the crystallographers, we've divided and conquered large proteins because we wanted to get the structure of at least the business end of the molecule, the catalytic domain, the ligand binding domain, things like that. Things that were known to be important for drug discovery to move the project along. Perhaps the crystallographer tried to crystallize something bigger and then said, you know what, I'm going to try a proteolysis digest and see what I get. And I got 3 different things. I'm going to try to crystallize each of them separately. And then they were successful. So we moved on and we determined those structures. Because these large domain proteins are difficult to crystallize, this was a tactic that we just pursued because it was working. So, and I guess I say thousands of structures have been determined that way. But now what's really exciting for the cryo-electron microscopists is when they tackle the intact protein with all the domains, despite its flexibility, and they finally get their first experimental maps, a good deal of that structure has already been determined by crystallography. So they can just get those PDB files and dock them into their electron density maps. And they don't have to do all the model building of those regions. It's already been done. So those structures come together very quickly with work that's already been done. And then this is the kind of the final story. For some projects, you can actually do both techniques in one lab, and that happened in the Evanston lab, in the Rosenzweig lab. I would say 15 years ago, there was a really, really brilliant graduate student in Amy Rosenzweig's lab who, in a heroic effort, solved the structure of a very large methane monooxygenase. It's a protein. It's an enzyme that takes methane out of the atmosphere. So it's something that could help with global warming, basically. And she determined this structure. It's a membrane protein. It was very difficult to do in that time. And in order to do that, to be able to be successful with crystallizing that structure, she had to use detergents to purify the protein and to solubilize it and to crystallize it. So the problem was that the detergents rendered that enzyme inactive. Nevertheless, it was a Nature paper. They didn't really, it was the first structure of this important, huge molecule that takes methane out of the atmosphere. So you can read what the abstract concluded with, that the new paper is an EM. Sorry, I'm getting ahead of myself. The new paper, the structure was revisited by cryo-EM. The students that worked on it did an analysis where they studied some lipid nanodiscs that were more native to the membrane that these molecules were purified from. And they found that the detergents that had been used in the past caused this to happen in the activity. The activity went down, the copper binding went down, and they concentrated on the lipid mixture that gave the most activity. And they made cryo-EM grids out of that. And what they saw was these lipids in the structure. They saw them on the outside, and they saw them on the inside of the structure. And these lipids that supported enzyme activity, they explained a few things. They explained the activity increase, that was obvious, but there was also a third copper binding site, this one, that was not occupied in the structure, the crystallography structure that was solved in the presence of detergents. But this third copper was present in the EM structure. That may have explained the increase in activity or the decrease in activity for the crystal structure detergent solubilized protein. But in any case, that wasn't the only thing, was this binding site for the copper. They also saw a couple of areas. This is just one highlighted in the paper. This cyan loop here was something that was disordered in the crystal structure. And it makes sense that it became ordered in the cryo-EM structure because it's surrounded by those lipids that were added to get from the nanodiscs to get the cryo-EM structure. Okay, so crystallography and cryo-EM are really exciting techniques for me, at least. I hope I made that clear. They let us see our molecules of interest. They let us see what's hydrogen bonding to what. Water structure is very cool at high resolution. All sorts of things are cool at high resolution. But actually being able to see something rather than just see how does it work. I have this proof from this assay and that assay. You can take that data that you have and then see if it is explained by the structure. And then you have a complete story, and that's very exciting. So cryo-EM is definitely important for the larger and larger molecules, and will be continuing to grow exponentially for the next foreseeable future. Although crystallography still has the highest resolution and structures can be solved usually more quickly, the highest resolution isn't always necessary. The last thing I say here is the resolution that you need is only that which explains what the question you're trying to answer. Although people do like to brag about having a hole in their phenylalanine ring density. And then again, just contact us. I've got my email address here. It's just my last name at Northwestern. And we'll take any questions you might have. And this is what all your amino acids look like. It just didn't fit where I had it before, but you can see the electron density looks like your amino acids, like you were taught in school. Anyway, thank you for coming. Oh, yes, sure. No, we don't. I think it's now it's a separate facility, but I can put you in touch with the facility manager. I think they're part of IMCIRC. They used to be separate, but now I think they're part of IMCIRC. Do you know what IMCIRC is? It's in Evanston, but it's I don't either. I used to know what the acronym meant, but I've forgotten. M stands for molecule, though. There have been some people are using glutaraldehyde to, yeah. That's one thing. Yeah, I think it's probably tricks with whatever else is around your stuff in solution that might get them to stick together. But also, you could try fusing them with molecular biology and try to get them to stay closer together that way. No, that's definitely something you could get advice about. But you brought up a really good point that I forgot to make, which is crystallography requires higher concentrations and more material than cryo-EM does. And you make a really good point about the lower dilutions. In order to get your single particles well separated and be able to collect images in EM, you have to go to lower and lower concentrations. And that's another one of the variables that you figure out when you're making your first grids. Is my concentration too low, too high, just right? With protein, we tend to try to go for 5 to 10 mgs per mil, and it's maybe a 10th to a 100th of that for EM. So yeah, thank you for the good questions.
I have a question. I don't know if you can hear me.
Yes, we can hear you.
Okay, wonderful. So I realize that you're not able to utilize paraffin embedded tissues for cryo-EM. Is it possible to re-embed them? I read somewhere that it's possible to use them.
Did you say parafilament?
Paraffin embedded, like if you have bone marrow biopsies that are
Yeah,
embedded.
I believe you might be able to go to the Nuance facility. We don't allow polymers and stuff in our microscopes at this time. So I believe that might be considered some kind of a polymer that could degas. Is that correct, or am I wrong?
I know that you're not because I guess you could develop crystals in the process and that could interfere with the interpretation. So, but I read somewhere that you could re-embed those tissues in a different resin type that may be more suitable for cryo-EM. And that's why I was asking if it's possible.
Well, it may be, but also tomography is possible, but we're not doing it yet in our facility. But um, if you don't have to embed it in a resin, is it possible to just use
Fresh tissue.
frozen slices or something like that?
Yeah, I guess we could obtain a couple of frozen material and utilize those.
But you could definitely come and talk to us about it. I know our microscope stages tilt plus and minus 70 degrees. So we're able to get a lot of different angles on tissues, but no one started that kind of work yet in our facility.
Okay,
And I
well,
do know that
we
the
should
Nuance
chat.
Center, though, does some of that. Nuance does.
Okay, thank you.
Yes? Yes. Yeah, so the old-fashioned way of keeping your cryogrids protected and frozen in long-term storage dewars was to use a 50 mil conical tube with a long string on it. You're shaking your head like you know what I mean. It becomes a terrible disarray of strings all tangled together. So the EM people, like with many other parts of the science, have learned from the crystallographers and moving to a puck system. So the puck system is like heavy metal pucks with. Okay, so we're getting a couple of those systems for the Evanston labs because there's one lab on the campus, and then there's the new lab with the Krios is going to be downtown in a new building. So in order to transport samples from place to place, so the pucks can be put in a storage cane that goes in a big dewar, but then there's a cane that holds 10 pucks that also fits into a dry shipper. And then that can be transported to the other locations. That's how people also get things to national centers. But we're ordering those systems in the next week or so. But the Chicago campus also got one or two of the transport size. So they don't have the large, I don't know if they're going to get the large storage canes for the long-term storage dewars, but they are getting the transport dewars with the 10 canes and the shipping cane. I'm not aware. Would you ship it in a dry shipper with? Well, FedEx is the proven method, I guess. I don't think this. Luda, do you know? But do they take dry shippers too? Yeah, I don't know. I just know that a dry shipper is meant to be shipped dry. So you make sure it's cold. You have to dump out the liquid before you send it to FedEx or to any courier. They won't take it if it has liquid sloshing around on the inside of it. There's a sign up in Nucor. If you have not been trained yet, you can become trained. Or you can send your samples and have our facility manager, our specialist, screen the samples for you. It all depends on what you want to do. If you want to become a microscopist, then by all means you want to get trained. But if you just want the answer to know how to make more of your grids, then you can just ship them and ask someone in our facility to screen them for you. It's being delivered end of February, 1st of March. How long it will take before it's commissioned and online, I don't know. But yeah, but it is coming. But we're getting nice data from the glacios. Whose lab are you in? Oh, well, welcome. Oh, great. How are you freezing your grids? Is there a Vitrobot too? Oh, great. Well, we have 2 Vitrobots on the Evanston campus that you can also use. Especially, it sounds like you already have experience, so you can walk in, talk the talk, and you'll get the stamp of approval. Oh, okay, that's great. Yeah, I don't really know what's going on here. I just knew about the glacios that the Chicago campus is getting because of this storage system, this money that we were given for the storage system. Yeah, that takes a while. Where is it going to be located? Oh, in this building? Oh, that's great. Very cool. Thank you.